Well, greetings, my friends, and welcome to another lesson in the pharmaceutical R&D process. Today, we're going to be talking about the second half of biotechnology drugs. Those that are based on nucleic acids or DNA, RNA, or what in a more larger context can just be called genes. Now, before we jump into this, we want to spend a few minutes talking about some basic properties, if you will, of DNA, that's what's called a nucleic acid. And so one thing we all probably know is that DNA is a blueprint for cells to function, that is to make the machinery that keeps cells going, for cells to reproduce, okay, make daughter cells of the same type, and also for an entire organism to reproduce. So for a human being to reproduce, DNA is part of the coding for that. Now, interestingly enough, it's only a very small part of the DNA that actually contains information that's directly encoded into proteins. Somewhat less than 2%. That's not to say the rest of it is what has in the past called junk DNA. They're finding new purposes for the other 98% every day, some of which probably has to do with regulation and folding of the DNA molecule. Now, one very important thing to keep in mind is that the DNA in every cell of our body is the same. And the reason we have different cell types is because only certain genes within the DNA of a cell are expressed, okay? But it's worthwhile keeping in mind that all cells have the same DNA, and that DNA is essentially the same from the day you're born to the day you die, except for an occasional mutation. This is why a company like 23andMe or Family DNA can do a DNA analysis, and that can be good for life. I just drew this slide in here to show you that, to illustrate how DNA is very similar between people. It's about 99.9% identical. And I just had to throw in this personal slide. This is my wife and I. Here's my dear wife from many years ago when we had our two little girls. Now, they're not identical twins, but they're very similar. So back to the role of DNA and its importance. Now, the interesting thing is only a handful of rare diseases result from a defect in just one gene, okay? Sickle cell anemia, Tay-Sachs disease is another example. Huntington's disease. They're relatively rare. Most diseases that we're familiar with, Alzheimer's, diabetes, cardiovascular, cancer is somewhere in between these two things. But most of these, what we call chronic diseases, result from a combination of genetics and environment. So you can have an identical twin, for example, an example might be schizophrenia. Okay, there's a fairly high concordance between identical twins, about 50%, but yet one identical twin can get schizophrenia where the other one doesn't. So clearly it's not completely a genetically defined diseases. So just keep that in mind, that because this is important when it comes to therapeutics, going in and fixing one gene for most cases is not going to solve the problems of chronic diseases that, really burden society. And then the other thing I wanted to make a point to mention before we go on is that consequences of genetic alteration, that is messing around with the DNA. Well, if you do that in what's called somatic cells, cells of our body like muscle cells, T cells, the changes that occur are passed only onto that cell line Whereas if you do that, if you make a change in germline cells, egg or sperm in a person, those changes are passed on to the offspring. Okay, those become, in a sense, permanent. So we want to be very cognizant of that when we're talking about genetic therapies. And again, not to overburden you with strict science, but another very important thing to just keep in mind when we're talking about genetic therapies is that there is a particular flow of information that occurs within biological systems, and that has been defined by Watson and Crick as the central dogma of biology, and that is pretty much that information flows from DNA to RNA to protein. Okay, so you have this molecule of DNA, double-stranded DNA, which ends up making it's transcribed into RNA, okay, and RNA then is converted to protein. It doesn't happen the other way. This is the direction that normally occurs, with some very rare exceptions that we're not going to talk about right now. So let's get down to what does this have to do with biotechnology? Well, there are many different forms of gene-based therapeutics, and I'm just touching on a couple of them here. All right, so I've stuck with these definitions. One being cell therapy, okay, which we can consider augmenting functions of existing genes. Gene therapy, which is adding a gene that works to a person who has a gene that doesn't work. And then gene editing, which is repairing the gene that doesn't work. Okay, so again, I want to point out that these terms are rather loosely defined, a lot of overlap. You may find different definitions, you may have them categorized differently in different contexts, but it works for our purposes. So let's start with something called cell therapy, right? In cell therapy, typically what's done is cells are removed from the body, something is changed about them to improve their function, and then they're returned. And one classic example is use of cell therapy in cancer. So let's take a look, go quickly through this diagram. So blood cells would be removed from a cancer patient, okay, and specifically the T cells, the type of immune cell, is isolated. And then what happens is a gene which codes for a particular receptor, chimeric antigen receptor, CAR receptor, is inserted into the DNA, which is in that T cell. So the chimeric just means that it's a combination of the native gene and some outside gene. Now this outside gene is something that is produced in a lab. It's an artificial type thing, but it codes for a receptor that is on the cancer cell. Okay, so now when this immune cell that has these receptors, okay, which has been artificially added to it, that is grown, allowed to reproduce in a lab, and then infused back into the patient. Now these T cells they express this receptor because they have the DNA that codes for it. We've added that. Now they can attack the cancer cell. So you can see the cancer cell has the receptor, and then the CAR T cell has the what you might call the co-receptor that binds to the cancer cells and then ends up killing it. Because going back, you look at the original T cell. Well, it's a kind of a killer cell. It helps protect the body, but without having those receptors, it doesn't recognize the cancer as the bad guy. Okay, so CAR T cell therapy works kind of that way. Again, I'm just trying to give a simple overview of how these things work. They're obviously very, very complicated. Now let's take a look at what we call gene therapy. This is a situation where you have a gene present, but it's not functioning properly. And what we're going to do is just add in the proper gene. So here's our patient here who has a condition known as beta thalassemia. So that is not an insufficient production of hemoglobin to carry oxygen. So they're always kind of cyanotic or not enough, don't have enough oxygenation in the blood. And on a molecular level, it's due to a lack of this particular protein called beta globulin or beta globin. So what do we do about that in terms of gene therapy? Well, start out by taking bone marrow from the hip bone, and that's harvested. And these are really just bone marrow cells here, which are cultured. And then here's where the magic comes in. What's added to the bone marrow cells in vitro in a petri dish is this interesting substance here, which is basically the gene of interest, the gene that codes for beta globulin. Okay, that's in yellow here. That's produced synthetically. But you'll notice we're not just pumping in the gene, we're putting in the gene within some other stretch of DNA. That other stretch is called a vector. And a lentivirus is actually a virus. Quite often adenovirus or cold virus, mild cold virus can be used. And the reason that this viral vector is included with the gene, and the combination is called a transgene, is because the virus has the machinery to allow this entire sequence of DNA to be incorporated into the host genome. All right, so the transgene is incorporated here into the host genome, and now you have the functional gene. So those cells are allowed to reproduce and infuse back into the body, and in principle, the subject can go on then and have the proper amount of beta globin produced and have the proper amount of hemoglobin. And in principle, they're cured after this point, because as the cells reproduce over the course of a person's lifetime, of course, the DNA has that vector, has that transgene in it to make the proper globin. Okay, so let's take a look at the procedure of gene editing. And there are a number of ways that gene editing can be carried out, but one of the most recently discovered techniques takes advantage of this system called CRISPR. Now, the letters underlying CRISPR are kind of complicated. I don't want to get into that, but the basic idea is that this enzyme system has been taken from bacteria because the bacteria use it to cut out viruses that have been incorporated into themselves through infection. So we are taking advantage of a bacterial system here that already exists. So let's take a look at how this might be used to help a particular individual here deal with a disease. So here is this gal, Susan, who has sickle cell disease, which is caused by a single DNA letter mutation, just a single tiny little nick or improper alphabet in a rather long gene that is very important, okay? But she's got a defect in that particular gene. So what happens is in the lab, we know what the gene sequence can be. So what's called a complementary RNA sequence is manufactured in a lab of what the proper are of that gene sequence, and then it is added with the CRISPR system. Okay, so in number 3 here we see that the that this CRISPR and these enzymes in the CRISPR system, basically what they do is they cut out the piece of genomic DNA that matches your guide RNA. So we've got that particular system put together, and sure enough, what it does is it cuts out the entire gene, if you will, or a sequence that matches the guide RNA. So now it's removed, okay? But that's not really solving the problem, because she doesn't have a proper sequence. And this is where the difficulty comes in. It says here, scientists insert a healthy DNA sequence to replace the clip sequence. CRISPR can do that for you, but it's really still something which is being worked out, and there's a lot of challenges to getting that done properly. So this is kind of a system with a lot of potential, but still being worked out on. And as it says, if all goes smoothly, Susan no longer suffers from sickle cell disease because that particular defective gene has in effect been repaired. Okay, so actually it's a very, very long gene incorporating hundreds, maybe even thousands of particular components, what are called nucleic acids, and a DNA bases, and just the piece of it has been snipped out and then replaced by the proper piece. So that's gene editing. And I want to just kind of summarize this by saying a lot of these techniques are in development. They haven't quite been finished up yet, but I would imagine in the near future this is going to have quite an impact on health. Well, one last thing I want to go over, although it's not technically gene therapy, is the use of RNA-based vaccines. These are what Moderna and Johnson & Johnson have developed to fight COVID infection. So let's kind of take a look at this. Well, we have this coronavirus, and as you can see, the coronavirus is kind of written as a cartoon here, but the DNA of the coronavirus and the enzymes, all the the parts of it, the working parts are inside this membrane. Okay, and outside the membrane of the coronavirus is what's called spike protein, right? Spike protein really is not infectious, but it helps this coronavirus particle bind to our cells. So what happens or what's done is that this spike protein, the amino acid sequence, is determined in a lab. Okay, it's a polypeptide, and the amino acid sequence is determined. Based on that, we can work backwards on paper and say the RNA that would code for that protein is such and such, and we actually can manufacture that RNA that would code for that spike protein in a lab. So that's grown up in a lab. We're not going to talk too much about the technology, but that's grown in a lab or manufactured in a lab. And then it's encapsulated in a lipid-like material, that like a fat-like substance, like this is what you might call a micelle, because that lipid-like material is necessary to get the mRNA into cells. So now the lipid-like material with the mRNA is put in a bottle, and this is where formulation comes in. It's very important to store these things under the right conditions so that that lipid-like material doesn't fall apart or degrade. And then it's injected into a person. Injected because obviously if you gave it orally it would just degrade in the gut. It's injected, and the lipid binds to a cell, and this can be almost any cell, and the RNA ends up in the cell. What happens then is the RNA is what we call expressed, okay, or converted to the protein, the spike protein, okay? So now our cells are manufacturing that spike protein. Now the spike protein, remember the spike protein in and of itself is not infectious. So that's floating around in the body after we took the vaccine. And what happens is the body produces antibodies to the spike protein. Okay, so now you've got a lot of antibodies. So when you're exposed to coronavirus, the entire particle, the antibodies are there waiting to neutralize it. And that's a little bit of an oversimplification, but that's how the process works. And one other thing that people often get concerned about is it possible, could it be that these RNA vaccines are messing up my genome? That's not going to happen because remember, we go from DNA to RNA to protein, okay? And we're dosing a person with RNA, it's only going to make protein. It's not going to go back to DNA or be incorporated into DNA. Okay, that's it for our discussion of Biotech Part 2. I hope it was enjoyable and that you learned something. God bless.